The overall goal of this protocol is to enhance axon alignment and myelination of dorsal root ganglion neurons using a static, pre-stretched cell culture system.
This method can help answer key questions in the nerve tissue engineering field, such as axon alignment and thickness growth. The main advantage of this technique is that this pre-stretch method not only enhances axon alignment, but also promotes myelination.
To begin this procedure, pour the mixture of base and curing agent into a tissue culture dish. Keep the gel mixture under vacuum for 20 minutes to remove air bubbles. Then, place the gel mixture in the oven at 60 degrees Celsius for overnight curing. After the PDMS membrane has cured, treat the surface with oxygen plasma in the plasma cleaning and etching system for 3 minutes. Subsequently, cut a piece of rectangular membrane from the dish. Fix it onto a stretching frame with the oxygen-treated side facing up. Then, place the frame on a stretching stage and evenly stretch it by turning the knob on the stage until it reaches 10% elongation in the longer axis. Next, fix the stretch by tightening the screws on the frame. After that, remove the frame from the stage. Ensure the membrane surface is dry and free of dust before placing a silicone chamber onto the membrane. Allow the sticky side of the chamber to attach tightly to the membrane. Then, sterilize the surface with UV for 10 minutes.